Resting state fMRI, this really came out because for a long time people were doing experiments where there were sort of on blocks and off blocks, and they'd always look at what was more active in the on blocks. But then people started noticing that when you looked at the reverse and areas that were sort of more activated during the off blocks, there were areas which struck people as a little counterintuitive at first. And moreover, they seemed to be somewhat consistent across studies. And so people started looking at this and found out that, in fact, there does seem to be some consistency in the patterns of brain activation that that we see when people aren't engaged in any specific tasks. So then the question becomes, what are you doing when you're doing nothing? So this isn't necessarily when you're doing a control task, but when you're actually just sort of resting, staring at a fixation cross, not doing anything. Right? And so if you're the Dalai Lama or if you've trained in meditation for many years, you might actually be able to sort of shut your brain down to the point where you're not really actively thinking about stuff, but you're still conscious. But for the most case, of course, we're not scanning people who have that sort of level of mental control. So people are doing stuff, and you would expect that people are doing different stuff, right? So given no particular task, if you're just sort of sitting there or lying there in the MRI scanner, and I'm going to open this up to you, what do you think you might be doing? Reading? Breathing, yes. That doesn't require a lot of brain function, though.
Mind wandering? You're just like thinking about like what you're going to do later or something?
Yeah, okay, so that's good. So mind wandering, but if we unpack that, so thinking about future events, what else goes on when your mind's wandering? Looking around, yeah. So you're in an MRI scanner, so there's not a lot of visual stimulation. You're basically sort of forced to see the screen, but yeah, you could potentially look around a bit and see the edges of the MRI scanner, that kind of thing.
Get really focused on the sound around you. So like the MRI machine is quite loud.
Yeah.
If you're wearing headphones.
Yeah.
And often when you're in it, you'd probably still be paying attention.
Yeah, so you tune into external sounds more, yeah.
I'm sure I get like really fidgety.
Mhm.
Start thinking about like even the smallest sensations, like tickles or like muscle aches or anything like that.
Right, and you're lying in this tube for like an hour, right? So you're going to start to become very aware of anything that's slightly uncomfortable or, yeah.
I ruined someone's data because the back of my head was itching.
Uh-huh, yeah. Itches, all that stuff, yeah. So that's good, a good sort of general coverage, right? So we've got sort of thinking about future events, becoming more tuned to basically sensory stimulation that is occurring, even if it's not intentionally sort of driven by the experimenter. Probably like self-talk, so actual language activity, right? You're sort of thinking to your thoughts to yourself, which employ language. So there is a lot going on, and there's probably, even if the actual content of what you're thinking about, next is different from what somebody else is thinking about, we can sort of broadly expect that people are maybe engaging in similar general categories of activity. Right, and so this leads to this theory of what is kind of the default mode of brain function. So when we're not sort of driven by external forces, what are our brains doing? Cognitively, what are we doing? And Binder et al., who really wrote the first paper describing these initial sort of consistent patterns of activation during these off blocks, suggested that it was, quote, adaptive processes, semantic knowledge and retrieval. So if you're thinking about things you know, that's semantic knowledge and retrieval. Representation and awareness, these are all big words that basically say what you guys already said. Directed manipulation of represented knowledge for organization. So that's like thinking is a more concise way of saying that. Problem solving and planning. So lots of stuff sort of going on here. And a lot around the same time, there was some other work that was done with PET scanning, not fMRI. And the difference is with PET, you can actually measure oxygen extraction, oxygen metabolism. And so in this study, Guznard and Rekel looked at the oxygen extraction fraction. So how much oxygen was being pulled out of the blood in different parts of the brain during rest. And this was an extended, like a 1 hour resting scan. And what they found, very broadly speaking, is that the oxygen extraction fraction was not consistent across all brain areas. So again, it's sort of suggesting that some areas are more active, they're demanding more oxygen at rest than at other times. One theory is that maybe these are areas that just have higher metabolic rates overall, But when we look at this compared to the fMRI results, it suggests that these are, again, the areas that are more engaged actually when people are at rest and not being driven by external stimulation. And so this went from sort of an interesting observation, phenomenon, sort of hypothesizing about what brain activity or cognitive activity it might reflect, to the study of resting state networks and using this resting state as actually an experimental paradigm rather than just sort of a one-off or sort of happenstance thing. And so in resting state paradigms, you literally just tell somebody stare at the fixation cross or lie there with your eyes closed, so there's no external stimulation for scans that last, typically about 5 to 8 minutes in length. And then you've got this whole time period. The challenge is that unlike fMRI, as we've talked about it so far, we don't have stimuli. And so we don't have anything to time lock to, and we don't have any predictions about what's going to sort of systematically vary in the brain as a function of stimulation, because there's no stimulation. And so the approach to data analysis is actually quite different, and it's typically correlating the ongoing bold activity over that whole 5-minute or whatever scan with the time course of activity in one particular part of the brain. So basically it tells you which other areas are correlated with the bold signal in that part of the brain. And so what you're seeing here over on the left is the arrows pointing to a seed voxel in the parietal lobe. So basically they identified one voxel. I'll talk in a minute about how they come up with these voxels. But you pick one and pull out its time course, and it's just going to be something that's essentially random, right, to the naked eye. But then you say, okay, take the time course of every other voxel, every hundred thousands of other voxels in the brain, and correlate them with that one. So instead of having some time course that's predicted by the timing of your stimuli, your time course that you use as a reference is just some part of the brain. And the map that you get, like this colorful one on the left, shows you the strength of correlation between every other part of the brain and that part of the brain. So here, where we've picked the seed in the left parietal lobe, you can see the big lump of yellow around that. That's not surprising because we would expect that parts of the brain that are right next to that voxel are going to have highly correlated activity because it's basically the same brain area. But then you can see across the brain, so the homologous area in the right parietal lobe shows high correlation, and then parts of the temporal lobe, this medial parietal area here, which is called the precuneus, some other areas. And again, this is consistent across individuals. So although this is actually from one study, not from one individual, but this is quite common if you choose that seed voxel in the parietal lobe or in the precuneus is another common one. Do the correlation, you'll get a map like this. And this has been called the default mode network. And the other part of it is that you see the sort of red, yellow areas that are strongly correlated with that seed voxel. But then there are other areas that are shown in purple and then most intensely in green, which are anti-correlated, meaning that when the bold signal in that parietal region is going up, those areas are going down. And when they're going up, the parietal one's going down. So they're basically sort of, there's a whole set of areas and network that's anti-correlated with this default mode network, so that there's sort of two things going on, and as one's going up, the other one's going down, and vice versa. Where they got these seeds from was really going back to those early studies and looking at those areas that seemed to be more consistently activated in the off blocks or the rest blocks during different tasks. And that was sort of the initial description of the default mode network, and it included the parietal regions and the precuneal regions, some frontal and temporal regions. So that was already sort of established. And so for doing these default mode network resting state correlations, now people just use those seed voxels, and it's pretty well established which ones to use just based on looking at other papers and what they've used, because people have come to agreement. So that's sort of one side, is looking at the default mode network. And you may wonder, like, what's the point? If we've already characterized this, why would we do any more studies around it? And where it's interesting is not really sort of within an individual or a group, but comparing between groups. So if you're looking at any clinical population, you could ask how the resting state network is affected relative to a healthy population. And people have started to look at that in some cases with stronger, some maybe weaker hypotheses. But the attractive thing about the resting state analysis is that there is no task. And so if you're comparing, say, a group of people with Alzheimer's disease with healthy controls, and you give them, say, a memory task, well, that's kind of interesting because they probably have deficits in memory. But at the same time, that means that they're going to perform less well, and they're going to find the task more challenging. And so once you see differences, you can say, well, these are in some way related to the fact that they have Alzheimer's disease, but are the differences we're seeing because they're trying harder? Is it because they're just not able to engage these areas and they just sort of give up, or what, right? So there's this confound between the, their task difficulty and the deficits they have and that sort of thing. Whereas if you just do resting state scans, nobody's got a task. And so everybody's sort of on the same playing field in terms of level of engagement. As long as you assume that even with Alzheimer's disease, people are still doing general sort of mind-wandering kinds of things. You could question that, but that's generally the approach. So it's more widely looked at in the context of sort of different populations, or you can do things, so people have shown that default network activity changes after certain activities. And so you can have people engage in some sort of activity and then look at resting state patterns before and after that activity to see if things change like that. So that's the default network. What's shown, on the other side, on the right side here, is that other studies have actually pulled out multiple resting state networks. So not just this default mode network and the sort of anti-correlated pair of networks there, but a whole up to maybe like 10 distinct networks. And they did this not by using the correlation approach that I described there, but using ICA, which we talked about a little bit in the EEG labs as a way of finding eye movements. But broadly speaking, ICA is what's called the blind source separation algorithm. So it's a way of, you can put data in and it'll find patterns that are consistent in that data and things that sort of co-vary together. And so again, you've got resting state data, you throw it in, and ICA will pull out a number of different patterns that are consistent, and you can look at how consistent they are across individuals. And in this one study that pioneered this approach, is what you can see. You've got on the left side the data from one individual, and then on the right side the data from the group, just showing that this might be a quote representative individual, i.e. the best one, but trying to make the point that this isn't just a group phenomenon, but you can actually find it in individuals. And so it pulls out the default mode network, but it also pulls out ones like this first one is very heavily sort of visual cortex based. The next one's sort of medial frontal and parietal areas. Another one is more frontal areas. But anyway, there seem to be a number of networks, and a lot of them, when you look at it, it's kind of like, oh, that looks like the auditory processing network, or oh, that looks like the working memory network. So it seems like even when these areas aren't deliberately engaged by a task, just the fact that they routinely co-activate in the support of particular cognitive functions causes them to have stronger wiring connections between each other. And then even at rest, whatever spontaneous activity is going on, or to the extent that people are engaging in, say, working memory as just supporting their ongoing cognition, these things seem to be involved. So that's a whole sort of area is just looking at resting state activity. And that is one aspect of what we would call functional connectivity analysis. So that's where we're looking at, basically functional connectivity is looking at correlations between brain areas using some sort of neuroimaging signal. You can do it with MEG or EEG data. It is sort of more interpretable if you're using something like fMRI or source localized MEG, where you can look at the different brain areas as opposed to sensor level kind of stuff that you might do with EEG. But broadly speaking, functional connectivity is about correlations in brain imaging data. And critically, it's not directional. So we don't necessarily know within, say, the default mode network, all those yellow areas, if which one sort of starts things going and causes other ones then to activate. So we can't really track the flow of information, just we know that these things are correlated. The other thing it doesn't tell us is if they're actually structurally connected, right? So you can imagine that two areas show correlated bold signal because they actually have direct neural connections between them. Makes sense. But if it's a network, you can imagine that information sort of flowing through the network from one spot to another spot to another spot. And so two areas might show correlations not because they have any direct physical connection, but because they're connected through some other areas. So they sort of send a signal to one, A sends to B, B sends to C, so A is correlated with C. The other possibility is that you've got something like the thalamus, which sends signals up to the cortex. And so you could have the thalamus projecting to multiple areas, in which case A has direct connections to B and C and D. And then B, C, and D all show correlations amongst themselves, but it's all because they're being driven by this one area rather than being directly physically connected in any way. So functional connectivity doesn't answer those questions for us, but at least tells us something about networks that gets us beyond just saying, oh, these different areas light up. And we imagine, in a traditional sort of fMRI analysis, we imagine that they might be part of a network because they all light up during this task. With functional connectivity, you can take that a step further and say not only do they all light up, but the actual time course of activation correlates across them, which suggests, is more strong evidence of them interacting. Yeah.
Sorry if this is a dumb question, but for reading the multiple RSNs, is this like from that MRI tech where you're looking ventrally to dorsal, and the higher the Z number, the more dorsal the cut? Am I thinking of that?
Sorry, the Z's numbers there? Yeah, so those are coordinates, and Z is the vertical dimension.
Okay, so
right.
But that was like, is that what you were saying before when you're looking like from the technician's point of view, where you're looking from their feet to their head?
Yes, so the negative values would be low, and the positive Z values would be higher. Yeah. So in these, like on the left, yeah, that's kind of funny because the left is all axial and then the right is the three different views. So on the left, it's sort of the leftmost slice is the lowest and the rightmost slice of the three is the highest in the head. Yeah.
Thank you.
Yep. All right. Another approach to looking at functional connectivity and correlations is called graph theory analysis. And here, typically what's done is rather than picking one seed voxel and correlating everything else with that, we actually look at pairwise correlations between, typically not every voxel in the brain, because that would be a huge number of correlations. If you've got 100,000 voxels times 100,000 things they could be correlated with, that quickly gets computationally heavy. And so instead, typically people identify nodes or sort of individual brain areas. So there's things like later today we'll talk about cytoarchitectonic maps, but different ways of identifying brain areas and defining those. So you might have a set of somewhere between 6 and 200 different brain regions, and you look at all the pairwise correlations between them. So you compute all those correlations, apply some sort of threshold, and only keep the correlations that are above that threshold. So basically you're sort of finding the strongest correlations and dropping everything that's kind of weak and maybe not statistically reliable. And so that's sort of the top thing in the image here, the top panel is that set of nodes that we're looking at correlations between. Then we look at the correlations. Here I've drawn the thickness of the line is supposed to represent how strong a correlation there is. And then we apply thresholding, which would mean we drop all the thin lines and just keep the thick lines. And then there's a whole set of mathematical routines that have been developed within this area called graph theory, outside of neuroscience, that look at how patterns of correlations operate and form sort of subnetworks and clusters. So graph theory goes back to like the 1800s and sort of basic math and applied math. Its probably biggest use right now is in analyzing social networks. So people have used this a lot just by pulling out like tweets and looking at patterns of who's tweeting and who's retweeting their tweets and how people are connected, or sort of friend networks and so on. And they can find that using that analysis, you can easily pull out like who's an influencer because they project out lots and have lots of people following them or retweeting them or whatever. So the same sort of concept can be applied to networks of brain regions. And here what we're seeing is that there's basically sort of two subnetworks, which we're calling Module 1 and Module 2. So Module 1, these different areas show relatively strong correlations amongst each other, but not really with other areas. And then Module 2, the same thing. You've got sort of a number of these things that we've colored in purple that are all correlated with each other, but not so much with the green network. Then you've got a connector hub, and then you've got within the purple network what they call a provincial hub, which is like within a network node that every other thing seems to connect to. And so it's a way of looking and sort of trying to describe that the network of areas that might show functional connectivity isn't totally random, but there's some level of organization. There's maybe multiple functionally defined networks even during a single task. Everything's not correlated with everything. There's sort of sub-networks that are maybe supporting different functions. And so you can take this in both directions, right? So you can use it in an exploratory fashion to just sort of characterize how the different brain areas are interacting with each other. And then you can start to say, oh, like this module 1, that all looks like areas that we see in working memory tasks. So maybe it's supporting a working memory component of this task. And then maybe those other areas are supporting more of a perceptual component to this task. And critically, if you can find something like a connector hub that connects those two networks, that might imply that that brain area is critically involved in mediating between the working memory functions of this task and the perceptual functions, or whatever it happens to be. So it takes us a lot of distance beyond just this sort of neo-phrenological approach of saying, oh, this brain area lights up and it does X, and this other brain area does Y, and actually looking at how the brain's organized into these networks and subnetworks, and which areas might be critical in relaying information between networks and that kind of thing. So those are all functional connectivity measures. Effective connectivity measures are ones that actually try and make inferences about the directionality of information flow between brain areas. So I said before, the functional connectivity ones, they don't tell us that information, right? They just tell us these things interact. There's some correlation between them. Effective connectivity is trying to look at a directionality. The maybe simplest and probably the most intuitive approach to this is something called Granger causality analysis, sometimes also called cross-correlation analysis. And basically what it does is so the typical correlation that you would be familiar with is what we would call like a zero order or lag zero correlation. So this is a time series, and we would normally just look at, from time 0 to time 1, what, is brain area A, is the signal going up or is it going down? And then, in brain area B, from time 0 to time 1, is it going up or down, right? And do that for sort of each time point and look at their relative correlations at the same time. What Granger causality analysis does is it lags that correlation. And that's what I'm trying to show with the dashed lines there, is you're saying, okay, if we take a reference area, so in this case our reference is the inferior frontal gyrus in the left hemisphere, Broca's area, and look at some other area, so the right hemisphere homologue, so the same area in the right hemisphere in this case. If we say, okay, the left hemisphere is our reference point, and we're going to say, okay, if we look at sort of its time 0 to time 1, does that predict what brain area B is doing from time point 1 to time point 2, or at time point 2 to time point 3? So we're sort of seeing if activation in area A predicts future changes in area B, rather than changes at the same time. And so in that way, you can make inferences about the directionality of information between those two areas, because if the left hemisphere IFG is predicting future changes in the right hemisphere, that suggests the left hemisphere is sending information to the right. Whereas if the right hemisphere is predicting what's going on in the left hemisphere, 1 or 2 time points later, suggests the information is going in the other direction. And so it's still a correlation, but it's just using these lags to determine which direction the information might be flowing in. The big problem with Granger causality analysis as applied to fMRI data is that we're not measuring neural activity directly, right? We're measuring changes in blood oxygenation. And I've already talked about the fact that these changes in blood oxygenation are caused by changes in neural activity, which cause glutamate release, which causes the glutamate to be absorbed by astrocytes, which recycle it, and then there's a whole chemical cascade associated with that, which causes blood vessels to dilate, which causes more oxygen to be present in that area. So there's a lot of things going on there that could have variable time courses. And so the big question with Granger causality analysis is, when we see these differences, do they actually reflect the underlying changes in neural activity and timing in neural activity? Or to what extent might they be affected by other changes, whether it's sort of plumbing, differences in how quickly the blood vessels can dilate, or any of the biochemical processes that are causing that vasodilation. So it's in principle makes sense, but once you start thinking about it, there are a lot of questions about how much stock we could actually put in those results. And so you still see papers, but there have been a number of criticisms published of it, and I think a lot of people are fairly critical of that. The same thing if you did it, say, with MEG data, might be much more believable because there you're actually measuring the neural activity more directly, and so the timing differences might be more plausible. But with fMRI, there are a lot of questions there. Another approach is called Psychophysiological Interactions, and this is one that you can really only do when you have some sort of experiment going on, so not with resting state data, because you really need something that's triggering some change in brain activity. And what you do is basically you have a time series from some brain area, which we're showing in blue there, and then you also know what the experimental design is, and you look at how basically for the periods where your experimental condition is happening. So say this is like, you can see this is sort of a simple on-off block design. You're basically looking for changes that correlate with changes in the actual task demands. And so essentially you're looking to see if the strength of correlation changes between the on blocks and the off blocks of the experiment, or between different conditions or something like that. So it's, and you can, typically you might use a relatively low number of different brain areas, but you can look at how that predicts activity in other areas. But it helps to understand the relationship between the experimental protocol and the actual network sort of correlations between different areas, whereas the other approaches have, that I've talked about largely are looking at that across time without respect to the experimental protocol. There are, even within functional connectivity, you can do a simpler version where you're basically just looking at functional connectivity during like the on blocks of the experiment rather than the whole experiment. But this gets, it's a little more mathematically sophisticated in making that strength of relationship. But a relatively underused or not heavily used sort of technique. And then the one that is of the effective connectivity approach is probably the most sophisticated and maybe the most well used in my non-systematic analysis of the literature. It's called dynamic causal modeling. And so with DCM, we're still doing this sort of effective connectivity where we're trying to make predictions about, does one brain area predict activity in another brain area? But mathematically, it's a lot more sophisticated. So first of all, rather than just looking at the time course of the bold signal, it's actually got a mathematical model in there of how, so first of all, it's based on, again, experiment-based sort of stuff, so not resting state, but things where you're actually manipulating stimulation or some task or something like that. And knowing when the task is going on, you make predictions about changes in neural activity. And then the model actually has a model of how changes in neural activity would affect changes in bold activity, which is kind of what we've talked about before in terms of defining the shape of the HRF and so on. But you can be fairly sophisticated about modeling that neural activity, whereas like with Granger causality, everything's done on the basis of the bold signal. Here you can basically sort of reverse engineer, take the bold signal and go back to make inferences about the level of neural activity. So that's one piece, is that it's a little more sophisticated in that aspect. And the other is that we're looking at correlations between brain areas, but in both directions. Rather than just sort of having one correlation value linking each brain area, so each node there is supposed to be a brain area, you've got one in each direction. And then the third thing is that rather than just computing correlations, you're actually comparing different models. So you're predicting or trying to predict how correlations are going to change as a function of your task, and you're comparing multiple models of what effects that could have. And so here we've got two different conditions. This was from an experiment involving a VR kind of experiments where you're navigating through a maze and you've got a first-person view, so that's the spatial navigation condition, or the same thing except rather than you actually controlling it, you're just doing like a fly-through. And so you're experiencing it. So visually, it's the same kind of stimulation, but you're not in control. So the question for that experiment was sort of what's different when somebody's actively engaged in the navigation process as opposed to just visualizing. And the areas involved were the hippocampus, so H, I think F is frontal lobes and P is parietal. I'm sorry, I've spaced on that. But the idea is that between those two tasks, similar brain areas would be engaged and generally in similar ways. But during the spatial navigation task, there was a stronger projection from the hippocampus to the frontal area. And so what you can do is you can have different models of not necessarily exact values, but how these different brain areas are connected to each other and the directionality. So in this case, you can see some of these connections are bidirectional, some are unidirectional. And you can compare models that have like bidirectional versus unidirectional, test them all against your data and figure out which model is most consistent with your data. So it's a lot more sort of hypothesis driven, whereas all the other techniques are really much more, let's just sort of pump it through the algorithm and see what comes out and then go, oh, that must be the truth. Here you sort of a priori say, okay, it might be this or it might be this, and we have reasons to predict these brain areas are involved, and we have a reason to predict that maybe parietal lobe is projecting to frontal but not the reverse, and then you can actually test that, and you can see if there's more evidence for that model versus some other model. So there's a lot more that's going on with DCM than with the other effective or functional connectivity measures, and that gives it a lot of power and why people are interested in it. At the same time, it's still modeling, and because it's so sophisticated, that also means there's sort of more degrees of freedom for things to go wrong. Right, so those models of how bold signal correlates with neural activity or how changes in neural activity would drive bold signal are models. And they don't necessarily take into account the fact that different brain areas might have, show different strengths of correlation between changes in neural activity and bold signal. They might have different timing features and so on. So it makes it, it's a model. So it's principled in some ways, but it's making assumptions, and those assumptions might be wrong. And in fact, there's some papers that have looked at this and found that DCM can, in some cases, make wrong predictions or not deal well with individual variability. So it's one of these things that sometimes just because it's so sophisticated and principled, people tend to believe in it. But once you start drilling down, you realize that there are some limitations to it as well. But that's kind of the theme of neuroimaging in general is nothing's perfect, and we're always making guesses and models and predictions. And we just need to keep doing it and get more data to figure out where we're right and where we're wrong.